Okay, we're sticking with the post-larval theme here, or larval theme for that matter. But so first off, I just want to thank this great team of people, colleagues and collaborators of mine, two of which who are oceanography folks. So in doing some of this work that I'll get to in a few minutes, it's really important, I've learned, to have a physical oceanographer friend because they can do really cool things and magic and stuff that's over my head. So we've been interested in a few different things, a couple of ALIs that we were successful in getting. One of them that's out in publication now is assessing the changing thermal preferences that egg-bearing lobsters have as their eggs develop. So as already been noted this morning, you've got this long protracted development period. And within that period, these females make choices about where they want to be. And we think that's because that helps drive potentially optimal hatch time. Hey, I'm supposed to be in this place at this temperature to hatch eggs. So we learned a lot from these experiments, which were primarily lab, but also field-based. And so what I'm going to talk about in the next few minutes is, based on where those larvae hatch, how is larval swimming and performance going to help them or hinder them in getting to those shallow coastal areas that Eric alluded to earlier? And then based on that, can we model how those trajectories look? So we're not reinventing anything here. We're simply just saying, hey, with some of this new behavioral data that we have in swimming performance, can we look at this? So we've been trying to use a combination of lab and field-based work to do all these things. And so I'm going to focus on these two studies today. So conceptually, what we're looking at is, we already know that climate change is making alterations to the Gulf of Maine, whether it's warmer or colder, mostly warmer. We're looking at the fact that populations and egg-bearing females, which were noted earlier, tend to be moving further offshore. And so what are the implications of that due to temperature? How are those larvae being impacted as they have to transit back to those shallow coastal, warmer waters? And so that's sort of where we're kind of putting our focus in terms of those swimming dynamics and the distribution of post-larvae. So I know, Eric, this is going to break your heart, but we actually raised larvae because, yes, we can't get them as robust as wild-caught larvae and post-larvae, but what we do have is the ability to have maternal history and to have controlled conditions, so we can really examine the history and the development of those larvae over time. So we know everything about them, including the moms that gave birth to them. So that was a really important attribute to this project when we're talking about post-larval, our source of post-larvae for this study. And so what we did was we reared these larvae. We reared them to fourth stage from multiple moms, and we reared them at two different temperatures. One was sort of a predictive model for end of the century temperatures in the Gulf of Maine, and then we used sort of an optimal, hey, we think they're pretty happy at 15 degrees. We use these are actually pseudochrysal tanks that were used for jellyfish and gelatinous zooplankton, so it gives them a natural flow. And we also had some controls that went right into the freezer that we chopped up and did all kinds of things to them afterwards. So we swam, we did 72-hour trials. We used a combination of animals that were swimming and post-larvae that were essentially couch potatoes, just sitting in tanks, just doing nothing. So we had that comparison, and then we filmed those over the 72 hours. We then dried them, we weighed them, we did total protein and total lipid to get a bit of a proxy of nutritional condition. And so this is just one video in particular that we did. It shows you on the left is the warm temperature, on the right is the cooler temperature. And so these animals are doing their thing. And then we noticed something really interesting 72 hours in, which was the warming temperature. Those animals tended to aggregate at the bottom more often. So the percentage of time they spent there not swimming was increased. And so what this looks like, if we plot all this out, we see that the mean proportion of PL swimming on the y-axis and the 72-hour trials on the x. What you'll notice here, a pretty stark difference between those two temperatures and the proportion of time that they spent swimming. And we express this as each replicate tank swimming per 2-hour time bin. We used a GAM model with binomial distribution to look at these factors here, and we did notice this difference. And so what we actually noticed was that at the warmer temperature, those PLs were swimming about 20 to 25% less than at 15. What I'm not showing you here is we did a cohort of wild-caught PLs as well. They had a similar pattern. They decreased by about 11 to 15%. So there was still a decrease in swimming at warm temperature. Maybe that's something to do with cardiac output. There's a lot of really good ideas that Eric brought to the table there. And so these are, things that we could be thinking of. A couple other predictive plots that we did for this study, looking at effects of temperature, the duration within the trial, the time of day. But I do want to point your attention to that plot on the left, which is normalized time of day. So the amount of time they're swimming throughout a 24-hour period. And there does seem to be a bit of a diel signal there, similar to what Harding found and observationally others as well. So we do think that there are times where they're just not giving it the full thrust that they have. And so that brings up the question of, well, what are they doing if they're not swimming, right? What are they doing out in that giant Gulf of Maine? In terms of nutritional metrics, there wasn't really anything that exciting that popped out. So except we did see a significant difference in mean lipid content between that warm swimming treatment we had and all the others. So there does seem to be some correlation with, hey, I'm swimming, I'm being taxed, and I'm gonna have to use more energy to do that. And this was also the case for the wild PLs that we used. So quick summary on this study, we did not see changes nutritionally. We did not see changes in dry weight or protein. Lipid content was reduced at those higher temperatures, at that 22 degrees. We also saw some evidence for maternal impacts on the effective time within PL stage. So this is only something you can get from hatchery raised animals, because we would take post larvae from a single mom as she was throughout that hatch. So we were able to see that as we took PLs that were later, that hatched later from stage one, or eggs that morphed into stage one, we saw a different profile nutritionally than early PLs. So there is some nuanced behavior, nuanced nutritional data as those PLs get older. Okay, so with that sort of eye on PL swimming and nutrition, we sort of asked the next question, which is, does this decreased swimming activity contribute to any of those differences in recruitment patterns that we're seeing? Or can it, right? So we wanted to model this offshore swimming as these animals are transiting inshore. So we worked with Jang Choi and Tom Littman from UNHCCOM. We modeled the effect. This is sort of like a mental exercise. This is by no means a comprehensive larval distribution model. It should not be taken with full authority here, but it is an exercise in, if we released larvae at these 4 locations from southern Maine here in New Hampshire seacoast, how would those larvae do? So we went into the literature, we took as much information as we had, empirical data from Stan Cobb, for example, on swimming speed. We used that diel information and dialed that in a little bit to this model. We had to make some estimates on larval duration and temperature and things like that. And we also considered sites that were at the 20-meter isobath as our minimal settlement areas. And so I'm going to show you a bunch of plots. I'm going to have to do this real quickly, but this is the key to those plots. So at the top there, that banner at the top is swimming speed. That was a high end that's reported in the literature. We dialed in diel swimming, so you could, 20% less at night, 10% less at night. And this bar chart here in the corner is how much percentage of larvae from each hatch location that are actually reaching the settlement location. The circles are the endpoints, and the squares are where the passive drift ends after about 10 days. Okay, so with that, what you can see in just a few iterations here is larval swimming and directionality. We also made that assumption that there is some directionality to their swimming. There's some data to suggest that from a few papers. And, so for example, at 18 centimeters per second, at a constant swimming speed, most of those larvae are going to reach some kind of settlement habitat, except for that last part, although there is some settlement occurring here. So you can get an idea with these different combinations what's going on, including no swimming at all, just if they were passive particles, including stage 4. Then we used the opposite. We went to the opposite end and looked at the 7 centimeters per second, which is also derived from some lab studies as well. And you can see that there's a lot of variability there, and that females that are hatching further offshore in general, and I want you to draw your attention to this yellow color as well. This is about 30 kilometers offshore. These larvae are just not getting back to where they should be. That was a big take home for us from this study. So it brings up the idea that we need to focus on behavior. We need those inputs. We need those thermal tolerances, and we need a better understanding of nutrition and nutritional state to understand how these larvae can progress through if they're in fact going to have to move further and have more distance to cover. So this was our first attempt at this. You can read all about it in this paper that came out recently. And with that, I will take any questions. Thank you.
Sorry. Hi, thank you. Great talk. I'm curious about your swimming behavioral trials and whether or not you specified different types of swimming behavior, like looking for prey or tortuosity or anything like that between the different temperatures.
All we did, I mean, to feed them their swimming trials, they were not. Can you speak into the sorry. Not used to standing back here. We did not feed them. We didn't do anything to them. We just let them swim. So one thing that we noticed was, and this was actually described by Herrick in 1895, was death feigning behavior. So we actually saw post larvae they looked dead. They would roll over on their back and just be swirling around in the tank. And then you'd poke them with a cable tie or a toothpick or something, and then they'd start waking up and moving. And we think and we saw this more at the higher temperatures, at the 22 degree. And we think maybe that's an effect of energy conservation, because those were the animals that had the lower lipid levels as well at the end of the trial. So there could be some behavioral, and that brings up a question, is if they are truly not swimming the whole time, what are they doing? Are they just neutrally buoyant and floating around? Are they trying to go down? Are they trying to probe the bottom and hang out down there? Spiny lobster post-larvae do that. Different system, but they're capable of resting periods in between their swimming. Now, they also don't feed at all, so they're only relying on their reserves.
Is there any indication as to how far a single juvenile could travel before finding a favorable bottom to settle on?
You mean a post larva? Yeah, post larva. Okay. Let's see. So Katz did some work in. There's a paper from 1994, and they did a modeling exercise from southern New England, so Rhode Island, offshore Rhode Island, into settlement locations into southern New England, and they had large distances that these animals were covering. I don't recall the actual distances offshore. I don't know what they're capable of doing. Maybe Eric has some thoughts on that, or others, but it's a scratch your head kind of like, when we did some preliminary trials in our swim tanks, we let them go for a week. It was like, how did we decide 72 hours? It's what we settled on after some really lengthy trials, and we just saw after a while, they just stopped swimming. So it was about a week.
Thank you, Jason. Mine is more of a thought process, and I'm kind of bastardizing yours and Eric's projects or presentations together. But getting to this thought that the lab-reared ones aren't quite as robust as we see in the wild. We've mentioned diet, but do you think there's anything that could be impacting just living in the dynamic, like the ocean, like whether it's the swimming or if they're constantly facing a current that makes them want to develop stronger abilities? So whether there's physical characteristics you notice between your ones that were swimming versus those that just got to hang out at the bottom.
So in other words, what were we missing, right, with our swim tanks? So I think, again, we brought in the wild caught PLs. I think that's important. We really wanted to do that to see, are they really that different? We know they're that different nutritionally. But the question we were asking, we standardized everything with these swimming tanks and a lot of the other metrics. I think in the wild, these animals are experiencing a huge laundry list of other variables they have to deal with, right? And so in reality, they are probably, Jesus Pineda published that paper about post larvae hanging out in rack lines, right? Floating weed lines. He postulated that might be a energy conservation mechanism as well, like hang out, kind of like a rest stop, hang out, fuel up, and then keep going. I don't know how common that is as a behavior, but or how often those things are set up in those elements exist, right? So I don't know. Yeah.
A really interesting data set. I'm wondering whether it seemed clear that the 22C got their nutrition done. So maybe they didn't have to feed anymore. Their tummy was full.
This gets into how we cultured them, right? So full disclosure, we did not use wild zooplankton to feed them. We kept them at one temperature, right, for the entire rearing process. That's just what worked for us. But we fed them pretty regularly, multiple times a day. We had somebody who worked in that hatchery every single day for 2 years. She had that was Abby Lemon. She had a few days off. I felt terrible, but we all chipped in, but it's a lot of work, right? It would be less work if we could just go out and grab a huge cohort of wild PLs and, we can have a bonanza. We can do all kinds of things, but it's just, that presents a whole other challenge in and of itself. So I was talking to Alex Asher earlier. I think the idea is in the future to be able to produce cultured larvae and post-larvae that are as nutritionally compatible. I think we can figure it out. It's just a matter of, you know, the effort and the logistics and the science is there for the most part.